So, do we have some students from UCL? Calmez-vous. Ça va bien se passer. So, the students from UCL will cheer even more for her as she is professor at UCL's Faculty of Pharmacy and Biomedical Science. She is also a distinguished and passionate cook. Please give it up for Professor Nathalie.
Compared to what we give as lectures at the university, so it will be in a completely different format. And so I hope that I will share with you some new data related to maybe a new world that you don't know yet, but I hope that after my talk you will know better about your own microbes. And so if I may have the first slide of this presentation, just to show you that I'm going just to invite you just to know what is in your gut finally, because you know that it is related to the gut microbiome. And so the next slide just to show you what I'm going just to talk you about. So finally, what is the microbiome? So in fact, you harbor in your own body hundreds of billions of bacteria, but also other microorganisms. And I will show you that they play a crucial role in host physiology and pathophysiology. And so I will just show you what are finally the dialogue which is established between those microbes and your own organs, starting in the gut for sure, but having a lot of effects elsewhere and at distance. I will just talk to you about what we call the dysbiosis, meaning alterations of this gut microbiome equilibrium that is linked to the health status, but also to some diseases. And I will show you also that finally there are maybe some hopes about this knowledge, just to develop new targets, but also maybe new drugs, new way of treating diseases or preventing diseases taking into account this microbiome. So I have the next slide. Yes, so, and this science is a science that often appears in the newspapers and magazines and so on. But as you can see here, finally, this science is really an up-level science, meaning that when I started my career, just to say that it was more than 30 years ago, when I started about this subject, it was very, very difficult to make it credible because there were not the techniques we have now in hands. But now we know that you can have in the well-reputed magazines related to science and medicine, you have a lot of papers which have been published on this topic. So it can be credible, but always we have to have criticism about this science. Next slide. The gut microbiome. And so I show you this slide. And so it is finally related to this iceberg. Finally, why? Because if I had given this lecture, I would say a few years ago, a few decades ago, it would have been completely different. Because to know what happens in your body in terms of microbes present in your gut, you only could have an idea of what was present in your gut by culturing those microbes. And we could only culture around 30% of the microbes you have in the gut. And so there were really restrictions on the knowledge in this field. And now there have been a big advance, which starts only just to show what happens inside the water finally, because there has been the sequencing of the gut microbiome. So you know that the sequencing of the genome, human genome, was quite important just to unravel new diseases and so on. And it is exactly the same for your microbiome, meaning the genome of your microbes. Because now we know better about which type of microbes are there, and a lot better about the functions of all those microbes present in our gut, thanks to this evolution of techniques. And so in my view, what happens now, the fact that we talk about this microbiome, I would say, in a lot of different contexts, that's because of this advancement in the techniques. And so if we analyze the microbiome by sequencing of the microbiome in all of us, so we all share very common features. So we all have common species, common genera of bacteria, common phyla for most of us, but we are all different. It is really our ID card. And so we can really be characterized by our own microbiome, meaning finally we are all different from that point of view also. And it may also explain why there are some differences in the response to treatments or prevention by diets, and I will come back to these later on. And so the techniques that are analyzing now, so it just shows you that finally, yes, you have to have an idea of the molecular biology techniques of the sequencing and so on, but I won't spend too much time on these techniques, simply to tell you that we talk about artificial intelligence, but also bioinformatics, which are very important tools to try to unravel how our microbes are present in our gut. And there are also a lot of progress that have been made in metabolomics, meaning some techniques that allow to show which types of metabolites are present in the blood, but also in the fecal material in this context of the gut microbiome. And those techniques now are applied to better unravel which functions of the gut microbiome are occurring. So it means that for sure to have the analysis of the gut microbiome, it's not simple. You have to have experts, but also interdisciplinary approach with this bioinformatics and artificial intelligence just to really understand what sorts out this. Next slide. Then when we are born, until what we know now, because it's really a science that evaluates quite rapidly. So we know that we are born normally without any microbes. So there are no living microbes in the placenta, for example, even if there are some controversies and some teams who try to show that honestly, so normally this medium is sterile. And it is only when you are born that you are in the first contact with the microbes. So it means that if you are born by cesarean, for example, so it means that you acquire the microbes that are present in the hospital. But when you are born, I would say, by your mother, by a classical way, then it means that the first microbes you are going to meet are the microbes of the vagina, but also of the fecal material of your mother. And so it really explains that finally the first microbes that are going in the gut of the new baby are, for example, Escherichia coli, which are quite strange. But what we know, whatever is the way you are born, it is really the first moments of the feeding, which is very, very important. When you drink the mother milk contains some galacto-oligosaccharides, that types of, I would say, undigestible carbohydrates that have as functions to feed this microbiome. And it is the reason why, as you can show here, so you have, I would say, a few days after being born, you have an increase of bifidobacteria, which are really taking advantage of the presence of this specific food present in the mother milk. And so after, for sure, so you will acquire a stable microbiome, and in the elderly, normally, it comes back to a previous status. Sorry. So previous slide, if it's possible, because it goes, yeah. So what you have to just understand it is that this microbiome finally is in contact with your intestine. And so depending on where you are in the level of the intestine, so you can see that finally we have also to fight the microbes to avoid them just to enter your blood, because it could be very, very dangerous. Sorry. And then that's the reason why in your intestine you have some goblet cells that produce mucus, for example. And this mucus protects you against the invasion by the gut microbes. But also we will see later on that finally those microbes can also produce some metabolites that can in then turn have an effect on the key cells present in the intestine. You have also endocrine cells in your intestine, and some metabolites produced by the fermentation of your gut microbiome. Those metabolites, like short-chain fatty acids, may activate the endocrine cells in your gut, lead to the secretion of gut hormones, and then have an effect at distance of the gut. Next slide. And then it is really a two-way organization. So meaning that if you have a stable microbiome for the moment, that's because your intestine produces some secreted immunoglobulins, for example. They secrete also antimicrobial peptides that can enter the content of your intestine and have an effect on the management of the presence of certain microbes. So you secrete some peptides which are able to manage the composition of your gut microbiome. And on the other hand, as I said, there are some elements from the gut that may have an effect on your host immunity, but also host metabolism. Next slide. And then it is a figure that just shows you, it's a quite complex one, but that shows you how you can have, for example, an effect of this gut microbiome, which by interacting with the food you are eating may have an effect at distance of the gut. So if you eat some complex carbohydrates, and you eat a lot of carbohydrates, you have to have the major part of your energy as complex carbohydrates. Some of them resist partly the digestion in the upper part of the gut, and then it can be used by some bacteria, which are called saccharolytic bacteria, and they produce short-chain fatty acids, acetate, propionate, butyrate. And what happens then? Those metabolites are able just to bind, to link to specific G-coupled receptors at the surface of your intestinal cells, including those endocrine cells, and it may promote, for example, the secretion of well-known endocrine functions, well-known hormones. For example, the GLP-1, glucagon-like peptide 1, is a very important hormone that regulates the secretion of insulin, but also regulates appetite and so on. And so, indeed, when you ferment in the gut, thanks to the gut microbiome, when you ferment dietary fibers in the gut, you overproduce those hormones, and then you have a control of the food intake, but also glycemia. So it's very important to better understand this dialogue. Yes, next slide. And then the question can come, and if it is quite stable when we are adults in good health, what influences mostly the gut microbiome? In fact, it is mostly nutrition. So there are several factors that have been studied in those two studies, for example. And so what has been shown, it is that among the factors that can shape the gut microbiome, modify the gut microbiome, even daily and in a minor way, the nutrition and the way you are eating is really crucial. And so it will be the best way just to try to keep your gut microbiome as important in your physiology and to avoid disturbances of the gut microbiome. Next slide. And then there are now some definitions of dysbiosis showing that finally, depending on what you are eating, it will have an influence on the secretion of specific compounds. So if you are eating a lot of fat and sugar and energy-dense food, for example, you have characteristics of the gut microbiome that finally allow the translocation of some harmful bacterial elements in your serum, and it creates inflammation thanks to the effect on the gut microbiome. If you are eating healthy with a lot of dietary fibers and so on, you will have a completely different way by which the nutrients will be taken by this microbiome. You will produce those short-chain fatty acids, and then you will have some potentially benefits from these interactions. And so for the last 15 years, on the left-hand side, I showed you that finally you can have a lot of pathologies which are associated with disturbances of the gut microbiome. And it is partly due to the diet, as I mentioned, but partly due also to the presence of disease. So even in pathologies like autism, for example, there are changes and characteristics of the gut microbiome that may explain the pathology. You have also some liver diseases like non-alcoholic steatohepatitis that is also characterized by changes in the gut microbiome, diabetes, obesity, cancer, and so on. So a lot of different diseases characterized by dysbiosis. Yes, next slide. And then I will just show you now in the context of obesity and metabolic disorders. So if you read the newspaper today, so you know that there are a lot of emphasis on the increase of obesity worldwide. And so that's the reason why I chose just to show you this context. Next slide. Then In obesity, it is well now described that inflammation is a very important process that may drive the metabolic alterations linked to obesity, like diabetes or cardiometabolic disorders, but also some psychological disorders associated with obesity. And inflammation is an important component. And for sure, the excess of adipose tissue may secrete some pro-inflammatory cytokines that may drive this inflammation. But there are also elements coming from the gut microbiome that may contribute to this inflammation associated with obesity. If I may have the next slide. And then on the left-hand side, you can see that when you have this dysbiosis associated with obesity, so you have a decrease of very interesting bacteria like bifidobacteria or short-chain fatty acids producing bacteria. You have an increase of some potentially harmful bacteria. And the result is that you disturb your gut barrier function. You disturb your mucus layer, but also the tight junction between your cells in the intestine. And as a consequence, there are some potentially pro-inflammatory bacterial components that may enter your blood and participate to inflammation. And among those, the lipopolysaccharides, which are the major components of the gram-negative bacteria, which are present in the gut, at a level is around 1 gram of lipopolysaccharides in your gut, and it can then enter in the blood and participate to the systemic inflammation. And then it is one of the common components that has been ignored for a long time, but we now know this story. If I may have the next slide. And then there are also, in addition to the lipopolysaccharides, some metabolites that can be produced by the gut microbiome and have an effect on the pathologies related to obesity. For example, so there are some bacteria, very specific one, like Klebsiella. When they are present in the gut of some obese individuals, it can produce the production of ethanol, of alcohol in your gut. And this slow production of ethanol may have an effect on the liver and then promote the liver disease. It is something quite strange that you can produce, I would say, enough ethanol in your gut, and that can have this effect. Another example, there are some metabolites produced from amino acids, for example, from proteins, and they are really known as factors like phenylacetate and so on that can also drive the metabolic disorders like the liver diseases. For the cardiovascular part, so it is mostly a metabolite called TMAO, or trimethylamine oxide, which is produced upon the collaboration between the gut microbiome and our liver that may drive the occurrence of all the cardiometabolic disorders. So there are a lot of metabolites, and as you can see, there are some metabolites which are really not so good, and that can be produced upon dysbiosis associated with obesity. Yes, next slide. How can we show that? And so I'll just show you one example of the type of data. It was performed in children, obese children. You know that obesity in children is really becoming very dramatic. And so they analyzed a cohort of obese children and obese children who presented some alcoholic, non-alcoholic steatohepatitis. And for sure, those children did not eat and did not drink any alcohol beverages, for sure. Next slide. And it just shows you that finally in the children who had a higher disease of the liver, there was an increase of the serum ethanol level. Not the level of ethanol that you can get when you are drinking alcohol beverages, but a constant increase of this ethanol that may drive this situation. It was linked to a specific type of Escherichia in the bacteria of the colon of those children. Next slide. This is the process for the production of TMAO, but in view of the time, I won't spend too much time on that. That's coming from the phospholipids, from some lipids you are eating in your diet every day. And so some bacteria may produce TMA, so which is a metabolite from these derivatives of choline. And our liver that receives this TMA transforms it into TMAO, and this TMAO is the actor in the cardiovascular disease. So it's really not only our gut which is responsible for this. That's finally a collaboration sometimes between what happens in our gut and what happens in our liver that may drive these pathologies. Next slide. So once again, you can also have an idea that this better knowledge of what happens with the gut microbiome, this better knowledge may also lead to new biomarkers. And so I don't know if you are already aware of that, but it's very difficult to diagnose non-alcoholic steatohepatitis. Very difficult, because you have to make a biopsy just to be sure of this. And so there are some hopes that some bacteria can characterize the occurrence of this disease. Next slide. Then, are there some hopes, finally, from this presentation? So can we think that we can change this gut microbiome in order to improve our health? Are there some hopes? What are, finally, the limitations of that, the gaps of that? And I will end my presentation by giving you some tools about this. Next slide. So here it is a representation that I find quite fun, I would say. So when you have a dysbiosis, it is just like you have disturbances of your grass, like you can see in the upper part of this slide. And then there are some different tools that can be used. So you can have what is called a probiotics. Probiotics, it means that you are going just to put some grains on your grass and to try just to let them grow and to restore by this way maybe the initial level of grass. So you have the prebiotics. Prebiotics is just like you give turfs. So you give some components that can help the right part of the grass just to grow and so on. You feed the grass finally, and it is also a way to do. Then you have what is called the fecal material transfer or bacteriotherapy. What you do, it's easy. So you skip all your grass and you replace by a new gazon. And so that's something that is quite strange. And then you have also sometimes antibiotics. And then you can maybe restore your health. Let's have a look to what happens. Next slide. Then it is the different types of tools that are available. And so going from the left to the right-hand side, so you can see some very drastic procedures. Fecal material transfer, it means that you are going to transfer the fecal material and the bacteria coming from a healthy individuals to a diseased persons. And so it's really changing everything, like replacing your grass. Then you have the prebiotics. You are going just to feed and to give some food that contain nutrients that are able to interact with the potentially beneficial bacteria. And then you have an improvement of your gut microbiome and maybe of health. You have the probiotics, as I mentioned. You give very specific types of bacteria in very high amounts, knowing that those bacteria are able to play a role in restoring the key gut functions. And then you have other types of procedures. I won't have the time just to go on this. Next slide. And then in the field of diabetes, for example, if we have a look of what happens with the fecal material transfer, so there are some studies performed in some diabetic patients, and it was the first studies performed, I would say, 10 years ago. Now, it was in a very famous team in the Netherlands. And then they have transferred the gut microbiome coming from healthy individuals to diabetic individuals, and the control, it was giving to the diabetic individuals their own microbiome. It was autologous transplantation as a control. And so it was really impressive for the first paper published that there was really an improvement of the insulin sensitivity that lasted a few weeks after the transplantation. A lot of hopes, but it is impossible to envisage a transplantation of the gut material for all diabetic individuals. It was more a proof of concept. Next slide. If I may have the next slide. Yes. And so the other papers coming from the same team and other teams just were more, I would say, cautious, stating that, yes, finally, there are some individuals that respond and others that do not respond to this gut transplantation by improving the insulin sensitivity. And there are also some reversible periods. So after 6 weeks, so for most of the effects, they disappear, meaning that if you have just to propose these as a treatment, which is sometimes debatable, anyway, so you will have just to go for this treatment very often during maybe the whole life of the person. So it's more a proof of concept. Next slide. Then if we go to the prebiotics, is it possible just to propose some foods that contain nutrients able to play a role on your gut microbiome? Next slide. Yes, so the prebiotic concept, so I can make a short cocorico, it was born in Belgium, indeed. So it was in 95, and so I was really happy at that time when I was a young postdoc to participate to the first studies on this concept of prebiotics, and the concept is the following. So you have advantage to eat some nutrients that escape the digestion in the upper part of the gut, and thereby you can feed some bacteria that produce some interesting metabolites and have an effect on health. Yes. The different compounds we have been working on are inulin-type fructans. So you can find them in the chicory roots. So we don't eat frequently chicory roots. It has been extracted at that time as a powder. And so the chemical structure of the inulin is as such that's a polymers of fructose, in fact. And they are completely resistant to the digestion by the digestive enzyme in the upper part of the gut. But they can be used by specific bacteria that express beta-fructosidase. And the bifidobacteria, for example, they can produce these type of enzymes and use this inulin as an interesting substrate. Next slide. And so there has been a lot of experimental studies, and so in the context of, once again, obesity and diabetes and so on, in many models, experimental models of obesity, when you give this type of food, inulin, so you can have an improvement of the gut barrier function, of the gut endocrine function by the process I have explained before. You can have a decrease in body mass, fat mass, and also improvement of insulin sensitivity, for example. Next slide. And so what we have done is just to inform, so it's in French, this slide, because we have now a recipe book that contains finally what you have to eat if you want to promote your intake of inulin. It's not chicory root for sure, but if you have to eat leeks or onions or garlic or asparagus and for example, the Jerusalem artichoke, called in French topinambour, are the most important sources in our country. Next slide. And then what we know is that it's feasible, finally, simply by changing the way you are eating to change your gut microbiome. And here it is a study performed in the UC Louvain students, so it was in Louvain-la-Neuve. They came for 2 weeks in dietary interventions at the university restaurant, and they received such type of recipes and so on. They were obliged to eat this type of food. And so we were taking the fecal material prior and after the intervention. And after that, so they could come to the university restaurant but eat whatever they want. And so maybe to come back to their usual diet and so on. And in terms of gut microbiome, it's really quite interesting. It is that it is indicated in orange in these fields that, for example, you have after the intervention, after the 2 weeks of intervention by the food, you have an increase of bifidobacteria. But as you can see, 3 weeks after, when they come back to their normal food, it completely drops down. So it means that if you want to feed specific bacteria with an interest on health, you have to have constant intake of these type of nutrients. And as soon as you stop, it comes back to the initial level. Next slide. And so we also evaluated, and it was also a good collaboration with our partners of different universities here in Belgium, so from Erasmus Hospital, but also with the University of Liège. We had an intervention study with these type of nutrients, with inulin-type fructans, and also they received some advices to eat those foods which are rich in inulin and so on, as compared to placebo, where they received maltodextrin, and they also received some advices to have healthy food, but not particularly rich in inulin, for example. Yes, next slide. And then what we saw it is that also in this intervention study, in obese individuals for 3 months, you had an increase of these bifidobacteria and some very specific changes in the gut microbiome that did not occur in the placebo group. And so this was really very specific changes in the gut microbiome. Next slide. But when we had a look to the biological outcomes, the change in body weight, in glycemia, in cardiovascular disease risk, and so on, there were some significant effects in the inulin group, but also some effects in the placebo. But what we observed, it was really a huge inter-individual differences in terms of response towards the dietary intervention. It's just like all individuals did not respond the same way. And then we can ask, why? What are the components able to explain this? Next slide. And then we went to some hypotheses. Is it the drug treatments that they are taken together that may play a role? Is it the physical exercise that was quite different between the individuals? And was it the gut microbiome? Next slide. And then concerning the drug treatment, so there is, as you already know, or you will know, a very frequent drug that is prescribed in diabetes. And so these drugs, in fact, is metformin. And metformin, in fact, when it is given, so it really changes a lot the gut microbiome, just like dietary fibers. It's maybe one of the components of the effects of metformin. So metformin effects, for sure, the effect is well described. It's quite efficacious as a drug, anti-diabetic drug, but they also change the gut microbiome in the good way. And for that reason, so for the people who were treated with metformin, there were no further improvement when they changed their diet. Metformin was enough to do the job to improve the gut microbiome. Next slide. And then there are a lot of studies with different dietary interventions showing that indeed, when you try to change the diet of individuals in the context of obesity, diabetes, cardiometabolic risk, there are really subpopulations who are really well responsive and others which are not responsive. And there are a lot of studies showing that the gut microbiomes may explain this. Next slide. And then we did the job with this intervention study, and we could see that there are some very specific bacteria that if their level is high at the beginning, prior the intervention, like Akkermansia or Butyrococcus, for example, the persons who had these characteristics of the gut microbiome had a better decrease in the body mass index upon the intervention. And when they had the high Coprococcus, a very specific bacteria, they had a better improvement of mood linked to the intervention study. So yes, there are maybe some very specific components of the gut microbiome that may explain the effect on health. Next slide. And the physical exercise also. And so we were really surprised. We didn't expect this, but when we classified the obese individuals into sections of peoples who really had better exercise, exercised a lot and so on, so there was a much better improvement of the health in this context. Next slide. And then there are also some probiotics approach that may play a role. So next slide. And so the probiotics, it is not giving a food and so on, but it is to give, in fact, very specific bacteria. And there are a lot of probiotics that are already on the markets. But to be honest, when we have a look to all the scientific data, there are not a lot of success when just giving existing probiotics to obese individuals to try to normalize their body weight and metabolic alterations. Why? Because those probiotics, those bacteria that are on the market have not been created in the intention to treat these patients and having a physiological effects. Next slide. And then there are some innovations in the probiotics field. And so there is one of my colleagues, Patrice Cani, so who, for example, just elucidated that one bacteria called Akkermansia mucinifila was a bacteria that was down-regulated in the obese individuals. And so it had an effect on the regulation of glycemia. And so he developed, in fact, with his team, and so with a spin-off, now he's out of the spin-off, but he was at the initiality of the concept. He developed the concept that if you develop new probiotics based on the fact that you know the bacteria that are missed and also you know the role of those bacteria on the metabolism, you can maybe have an effect that can contribute to the improvement of obesity. Next slide. And so it is really a study that is called Microbes for You that has been done. It's only a proof of concept, and to be honest, we always say, and Patrice is saying also, we will never cure obesity with a single probiotics. It's only a way to try to have an adjuvant to the effect, the metabolic alterations linked to obesity, but you will never cure the obesity. And it's really sometimes quite difficult when you have some journalists that can promote this type of products saying that you are going to cure obesity. Finally, it kills science, and it's not the objective. Next slide. Then the take-home message, just to say that finally, I hope you are convinced that the microbes you are in your body, so that you have just to be gentle with them, and so maybe to feed them in a nice way that you can keep them at the update of the physiological effect of those microbes. And so there are a lot of mechanistic studies that must to be done, and so there are a lot of research that must be done to try just to better understand why we are so different finally in terms of microbiomes and how it can have an impact on the way that we can respond to dietary interventions, but also drug interventions in the futures. Next slide. And so I would like to thank you for your attention and to thank all the people in my lab. Thanks.
This really interesting conference. Now, does the audience have some question for her? Yes.
Hi. Oh, is it the microphone working?
Yeah.
So thank you for your talk. Firstly, I just saw one point that interested me because I saw briefly mentioned something about antibiotics. So I take it that antibiotics might very negatively affect the gut microbiome if you take them. So would you recommend, based on the research that has been done already, would you recommend some sort of fecal transplantation from your own microbiome after you take antibiotics? Would that be a good idea?
So I agree with you, and all the data show that antibiotics for long-term treatments with antibiotics, especially in the early age, so in young children, it's really problematic in the way that the gut microbiome goes on later on in life. And so it's now starting just to be studied in order to know that there are consequences later on in life. But I wouldn't be in favor of proposing gut transplantation in this case, even if it is for a chronic situation that occurs there, because it means, as I showed you, that you really need just to have a constant procedure that must be done every week if you want just to combat at least the metabolic alterations. And so there are for sure some very severe infections in the intestine, and so that can be cured by the transplantation, but that's because it was really a critical situation. So I won't go for this, but there are new hopes of creation, in fact, of recomposed microbiome with some specific bacteria that can be given together and reconstitute, I would say, a pseudo-microbiome that can be given to the individuals. It's better and maybe less dangerous. What do you have in your gut microbiome itself? You have viruses, you have for sure some moods and so on, maybe dangerous, huh? Yeah.
Before going on to the next question, so you told us you were a food lover, you're a great cook. So can you tell us a little bit more about how you would choose and how you advise the people to choose what kind of food they can eat and what kind of food they can cook?
Yeah, okay. So what I would say it is that for sure all the advices, when people say you have to eat 5 vegetables or fruits per day, it's true. But we have maybe here a new way of thinking about this advice, because you have maybe to make some choice in the which type of food. So it must, the intake of dietary fibers, for example, is very important to take into consideration. So we know from dietary recommendation we have to eat 30 grams of dietary fibers, and we find them in these vegetables and in these fruits. And there are some specific vegetables that contain specific types of fibers that are more interesting than the others. Some types also of some cereals are more rich in some of these prebiotics as compared to the others. And so there are some, I can give you some recipes if you want, and to transfer this type of recipes to the audience, but the vegetables I have mentioned, so it's really, in French it's topinambour, because Jerusalem artichoke is less known, so you have also the garlic, onions, but also salsify, for example. It's very rich in this type of fibers.
So mainly all the names you mentioned are kind of all type of vegetables.
Yeah, that's true. That's true. All types of vegetables, and also in the project Food for Gut we have conducted, it was also important just to have vegetables that are cultivated in Belgium here, and so that's also local foods that can contain these interesting fibers.
Locally sourced, sustainable. So not only it has a good effect on the planet, but also a good effect on your guts,
so.
right?
You got it.
Okay. There are certain brands of yogurt that are promoting some really nice effects if you eat them or drink them every single day.
Mm-hmm.
What do you think about those?
Yeah. So in fact, the science related to probiotics I have explained a bit. So it also comes from studies in which there are some yogurts in which, or some preparation, liquid preparations in which some specific species have been prepared and so on and kept in a nice way. And so there are some intervention studies in some that are published in very good journals with a peer-reviewed journal showing the efficacy in very specific context in this. But the problem is that, in my view, that there is an organization in Europe, I don't know if you are aware of, which is called the European Food Safety Agency, EFSA, that is controlling the claims that can be put on food. So you cannot put a claim stating that you will be healthy and so on without having the proof that it is true, in fact. And so we are protected from that point of view. But there are a lot of, I would say, of problems in order just to accept those claims related to the gut microbiome. It may change in the future. It is a young science.
So I will not become handsome just by drinking Yakult, right?
No. You have just to move and to eat well and just to think well also.
Which is driving me to the next question. The first step for you for a healthy body is a balance between obviously what you eat, but also how much you move, right?
Sure. And so in the context of obesity, and that's the reason why I state that, so you will never cure obesity with a single intervention. So it must be multidisciplinarity. And so it's also important that the future medical doctors you will have to dialogue with also the psychiatricians or psychologists, but also the kinesists and so on, because it is really, and for sure dietitians and nutritionists, because it's very important that you are aware that we have just to have those patients around us, taking into account all the components of health. Yeah.
So, ladies and gentlemen, we are reaching the end of the conference. Do we have other questions from the crowd? Please raise your hand if you do so. Nobody? One question. Can you please stand up and speak really loudly?
Yeah.
So is there an effect? Is there a bad effect on ingesting probiotics?
Exactly. So for the general population, so what is well described, normally the probiotics you will find on the market in Europe and not on internet and so on, normally they are controlled in order not to have a negative effect. So first thing, but it doesn't mean that it is efficacious, okay? But normally it is allowed because it has no harmful effect in the healthy individuals. For the prebiotics also, because there are fibers which are fermented by the gut microbiome. So it means that if you are fermenting something, so you will have gases and so on. So there are discomforts sometimes. But we had some studies related to this. Not all the people can respond to this increase in dietary fibers in the same way. So we have to listen to ourselves also when we are eating prebiotics and so on. But for the probiotics, if they are on the market, normally they are controls, but they are not toxic. And for prebiotics, you have just to hear yourself and hear your belly in order to know if it's convenient for you or not.
And ladies and gentlemen, that is the best way to end this conference. Listen to your belly. That is really the thing you need to remind. Ladies and gentlemen, please give it up for Professor Nathalie
Thank you.
Delzenne.